Yeah, hi everyone. So in the last class, what we have learned about different types of virus and like what is the basic structure of virus, like capsid, envelope, and the genomic material. And few of them are quite informative as well. So today it's a little bit more conceptual because today we'll discuss about virus life cycle, replication, and transcription. Okay. So, as in the last class we discussed that the virus, if we see the genomic material, right, it can be dsDNA, right. We have slides, but I just want to give you one recall, and another is the ssDNA, right. It can be dsRNA, and another is the ssRNA. So there are different category of this virus, right. Now the question is whether the life cycle for each of the virus will be same for all of these virus. Of course it's no, because the genomic material is different. That means, let's say it's single stranded DNA. If it, then the virus first should make the double stranded DNA, right, for its replication, and then from there it need to be transcription, and then followed by translation. And if we consider about ssRNA, it can be positive sense, it can be negative sense. If it's positive sense, so protein can directly produce from it. If it's negative sense, first it has to make positive sense RNA and then protein, right. So based on this, we will see different types of life cycle observed in virus. So we will start with one virus, that's the respiratory syncytial virus, or RSV. Okay, so RSV generally infect mostly the children. You can note it down from 0 to 5 years children, and the older people, as well as the immunocompromised individual. So what RSV does, it's the symptoms are quite similar like influenza. So cold, right, cough, and some lung inflammation, same, right. And it generally, the outbreak happen during the winter time, right, that you need to remember. But the most thing is that if you see the viral structure, okay, so let's say this is the virus and it has three major surface glycoprotein. I don't want you to remember each and everything, but just for understanding of the life cycle of the virus, it's quite important. Okay, so this is the F glycoprotein, this is G glycoprotein, and this is SH glycoprotein. So what's the role of each of the glycoprotein? You can remember, G is the attachment glycoprotein, attachment. Okay, F is the fusion, and SH is the delaying apoptosis. Delaying apoptosis. Why I am telling you all these things? Let's say, then you will be able to understand the function of the enveloped virus and each of the enveloped protein. Let's say the virus first need to attach with the host cell. Then first the G protein will come. G protein will help in attachment with the virion, followed by the F protein. The F protein will help the virion to fuse with the cell. And the SH protein, its role is delaying apoptosis. Let's say virus want to propagate inside the host. Now if your cell go through the apoptosis, then the cell will be dead, and at that time the virus or the virion will not get enough time to mature and to propagate, right. So the virus want your cell, whatever it's infecting, to be live, and then once its propagation is done, then it doesn't care about your cell, right. Okay, so the simple thing is that, let's say it has one attachment protein, let's say it's the G protein, and this is the respiratory epithelial cell. Generally it infects the respiratory epithelial cell. So we are writing here, this is respiratory epithelial cell. Okay, now we have one receptor called CXTCR1. You don't need to remember all these things. So the cell expresses some receptor. The main function of this receptor, it's the chemokine receptor. So it's not that the cells are preparing the receptor for the virus. Oh, virus come inside. Nothing like this. So receptor has some role, right? It's a chemokine receptor, but somehow the virus is targeting this one. Now what's happening, let's say this is a CH3CR1 receptor and the virus has the G glycoprotein. Now they will interact. Once they will interact, the virus will be fused with the host cell. So in this case you can see now the virus is first endocytosed, and then this is the after endocytosis within the endosome. What will happen, the F protein, because endosome is also membrane, right? The F protein will fuse. Let's say it's the virus virion membrane, it's the cell membrane or the endosome membrane. Let's say endosome membrane and this is virion membrane. This is the F protein in between. The F protein will pull these two membranes together and then they will fuse like this. They will pull the membrane to, and then the virus and the host cell will fuse together. As soon as it's fused, the genomic material will be released in the cytosol, right? The genomic material is the RNP, that means the ribonucleoprotein along with the negative sense single stranded RNA. If you see, RSV is belong to pneumoviridae family. Remember, RSV belong to pneumoviridae family and it's a single stranded virus having the negative sense single strand, right? Now as it has a negative sense single strand, it can't make the protein directly. So first it need to make the positive sense RNA, or so from here it will do the mRNA. It will make some mRNA, right? So positive sense RNA, and from here it will make some protein. So the virus use the host machinery like host ribosome, host everything, elongation factor, just to make their own protein. So once the protein is made, then the virus will make some kind of envelope kind of structure or vesicle kind of structure for its transcription. So in case of RSV or most of the RNA virus, remember the genomic material, that's the RNA, is not going inside the nucleus in most of the cases, but of course exceptions are there. But most of the cases the replication happens in some kind of vesicular structure that's located in the cytosol. So you can note it down that replication and transcription, and transcription occur in the cytosol, that you can note down in most of the RNA viruses. So the first thing, the virus need to attach. If I say this is the attachment is the first step. Second step is the endocytosis, once the virus attached with the host cell. Third step is the fusion of the virus and the host cell, and then the genomic material will release. And once the genomic material is released, as it's a negative sense single stranded virus, what will happen, from negative sense to positive sense RNA will be formed, right? And then from positive sense RNA some protein will be formed, and those protein, because the virus in virus genome, virus has its own RNA dependent RNA polymerase. So this protein means it has its own RDRP, RNA dependent RNA polymerase. So it will use its own RNA as a template and it will replicate itself, right? So let's say from negative sense, how it will replicate. From negative sense, let's say this is a negative sense. Now once it will make some positive sense, and from positive sense again it will make the negative sense. You got it? So from negative sense, just one complementary positive sense. From the positive sense, it will use the positive sense as a template and again use the negative sense, because negative sense ssRNA is the genomic material, right? So anyway the virus want this, and to produce, to make the protein, the virus need the positive sense RNA. So the step will be from negative sense, it will make positive sense first, then the positive sense RNA will be translated, and it has so when it's translated, that means the RNA dependent RNA polymerase is there, and then it will help in the replication of the viral genome mostly, right? Now let's say protein is produced. Protein means it's all the protein, enveloped protein. Let's say A, G, SH, everything is produced inside the cytosol. Then they will be trafficked, right? See the diagram here. Now everything is just transported to the plasma membrane, right? and the genomic material, whatever is the replicated things of the virus, let's say from one virus multiple virus will be generated, right? So now these things are going to the plasma membrane, and from plasma membrane through budding, right, virus assembly and budding, what will happen? Again some new virus will be generated. That's the role, that's the life cycle of the negative sense RNA virus. One example is the RSV. Here few things that you need to remember. First thing, that first attachment happen, followed by this endocytosis happen. Endocytosis means taken up of the virus by the cell. Then fusion between the plasma, maybe the membrane, maybe the endosome membrane and the virion, right, with the help of fusion protein. Once it's done, then the ribonucleoprotein comes out. As it's negative senses RNA, that means it has to produce the positive senses RNA, right? The positive senses RNA will make some protein, and with this the virus will replicate and transcribe. And then as it's making the protein, let's say all the envelope protein and all the protein that's structural or non-structural protein, those will be assembled here, right? Once it's assembled here, mostly the envelope protein, and then the virus will bud from the cell. That's the life cycle. Okay, now next, influenza virus. This is also negative sense RNA virus. This is a Paramyxoviridae. Remember the family, influenza virus belong to the Paramyxoviridae. Paramyxoviridae, right? That belong to influenza virus. And another special thing of influenza virus is the segmented genome. Segmented genome, right? So what's happening here? Influenza also has two kind of surface glycoprotein. Let's say one is the hemagglutinin HA, and another is the neuraminidase NA, right? Hemagglutinin means it's just attached with the sialic acid. You have to remember it, sialic acid. That's the receptor. Let's say this is the host cell, it has some sialic acid. First the hemagglutinin will go and bind with the sialic acid, and then the endocytosis happen. And what is the role of neuraminidase? Neuraminidase just chop off the sialic acid. Let's say the virus want to bud, right? Now in the budding condition, if the virus is still attached with the sialic acid, then the virus will not bud. So the neuraminidase will chop off, so cleave the sialic acid during budding, but not during attachment, during budding, right? Now see the influenza life cycle. First the virus is entering inside the host cell with the hemagglutinin interaction. Let's say this is a sialic acid, and this is the hemagglutinin HA. Okay, after endocytosis, what's happening? Inside the endosome there is some pH mediated changes. I don't want to go deep. So what's happening? The fusion is, the virion is fused with the endosome membrane, right? Then the 8 segmented genome, they comes in the cytosol, but it has 8 segmented genome. That's the most important thing. It's not one genome, 8 segmented genome. And influenza, remember influenza, there is one special thing that's called cap snatching. That means it produce, or we know that in case of mRNA, let's say this is the mRNA, in the 5' portion there is one thing called cap, right? The main role of the cap is it helps in translation initiation, right? So what the influenza virus do, it just steal the cap from the host. That's why it's called cap snatching. Okay, cap snatching, that means it's stealing the cap from the host. Okay, then what's happening here, that the genomic material or 8 segmented genome is coming to the nucleus. Okay, and here the mRNA synthesis and replication is happening. In all the RNA viruses, the replication is happening in cytosol, but you have to remember, in case of influenza, they are coming inside the nucleus, and then replication and transcription happen. Because the major reason I told you, cap, you can write down cap snatching. Isn't it? It's cap snatching mechanism. That means it uses host cap rather than its own cap. So it doesn't have any capping machinery. It utilizes the host capping machinery. Got it? So now once it's done, once the mRNA is produced inside the nucleus, what's happening? The positive mRNA, the positive senses mRNA are coming outside and they are making some protein, and this protein are assembled and finally it's budding. Right? Finally it's budding. So all the things are quite similar, but the things you have to remember, it has 8 segmented genome, and that's why it's quite complicated for transcription in the cytosol. So all the genomic material won't just go inside the nucleus, and inside the nucleus it utilizes the cap snatching mechanism. In terms of in the exam point of view, it's quite important. Cap snatching thing, they make the positive sense mRNA. The positive sense mRNA again come out the similar thing that happened in case of the mammalian cell. mRNA is coming outside, in the mRNA is making protein. This protein again accumulate in the host cell here, and from here the virus again bud. But just think how the virus is clever, like how it's decided that when only all the 8 segment of the genome will come, then only the packaging, then only the budding will happen. Let's say 7 thing is coming, the virus will not bud. Let's say 5 is coming, the virus will not bud. Only when 8 segmented genome will come together, at that time virus will bud. Just think and try to find out why so. How clever is this virus? Right? Next is the dsDNA virus. Earlier we observed the RNA virus. Now it's the DNA virus. One example is the adenovirus here, right? It doesn't have any like well-defined envelope, you can say, but yeah, let's see. No, sorry, adenovirus, it has some envelope. Okay, so it's attached with the host cell, right? It attached with the host cell surface. This is the receptor, okay? And once it's endocytosed, what's happening? They are fusing, right? And the release of the capsid thing here, and then as it has the dsDNA, right? It has the dsDNA. For its replication, it need to go inside the nucleus. In the nucleus, DNA replication is happening, right? And then some early gene expression is of course there, because from DNA some RNA will be ready, right? So inside the nucleus, transcription is happening, replication is happening, right? And then this genomic material is again budding. So here the thing is, here you have to make mRNA, because if the virus want to propagate, if the virus want the protein, so virus make the protein through hijacking the host cellular machinery, right? They use the host ribosome, elongation factor, everything. So what this dsDNA is doing, that means from dsDNA you have to remember, from dsDNA you have to make the mRNA, right? Just to get the protein. So inside the, that's why it must need to go to the nucleus. So inside the nucleus, some gene expressed early, let's say those are quite essential for the later stage of the virion propagation. Those are the early expression gene. Those will produced early, and then again they will come inside the nucleus and they will help in the overall replication, followed by the late gene expression, and then the virus assembly, right? That's the dsDNA life cycle. Next thing that we know, that's the HIV. HIV is a very clever virus because it generally attacks the T cell, like helper T cell, right? It binds with the CD4, and then it's let's say its binding is happening, followed by this fusion is there, right? After fusion, what's happening? The genomic material is coming here, and this genomic material, right, reverse transcription is happening, and then it's nuclear import. Reverse transcription means from RNA, right, it's making DNA. right? From RNA to DNA it's happening, and the DNA will be imported into the nucleus. And once it's imported into the nucleus, DNA will be integrated with our host genome. That means whenever your cell will be replicated, or whenever the gene will be replicated, that means the virus DNA will also be replicated. As its integration is happening, that's why it's very hard to get rid of HIV, right? So once it's integrated, it will of course make some RNA, it will be translated, and along with the integration, the virus will bud and the propagate. All the surface glycoprotein is there, they will finally propagate from the cell. The special thing is here, it make DNA from the RNA, and it will be integrated into the host genome. Clearing this thing is quite impossible. That's why in each step you can inhibit the attachment, you can inhibit the fusion, you can inhibit the reverse transcriptase inhibitor, you can add, but no approved drug that target the integration. So once it's integrated, how you can eliminate this? It's very hard, right? So the HIV comes with the retroviral family, where from ssRNA DNA will be made, right? And the DNA will be integrated. You have to remember the concept. Trust me, if I give you one full slide that's having all the information, it's meaningless. But you have to understand. If you understand properly, then all the question will be solved. Next, for exam point of view, I have listed here some of the virus and their host receptor, because virus, it has some envelope, let's say glycoprotein, and this is the host cell, right? It has some receptor. So this is the receptor binding site, RBS, or receptor binding domain, RBD. There is some, and this is the receptor. So for HIV, the well-known receptor is the CD4, right? For influenza virus A, the receptor is the sialic acid. It can be alpha 26 or alpha 23 depending on the human and the avian. For SARS-CoV, as we know, it's the ACE2 receptor. For rabies virus, it's the nicotinic acetylcholine receptor. For poliovirus, it's CD155 receptor. For Epstein-Barr virus, it's CD21. For adenovirus, this is the CAR. CAR means Coxsackie adenovirus receptor. For dengue, is the DC sign. So for exam point of view, you have to remember all these things, but also I am provoking you to think that what's the role of all the receptor if virus doesn't infect host. That's what I told you in the first slide. So the cell is not making the receptor for the virus or welcoming the virus, right? Of course they have some role. You have to find it out. You have to think about it. Right. So it has some viral transcription. Viral transcription means it's similar like that of the normal transcription observed in case of mammalian cell or in other organism. But few things that you need to remember. Let's say if it is the dsDNA. So we know from dsDNA the transcription is similar from dsDNA to RNA, right? dsDNA to ssRNA. That's quite similar. It's not that much difficult. So it's controlled by host DNA dependent RNA polymerase. So DNA dependent RNA polymerase is present inside the host. That means the adenovirus can use the host DNA dependent RNA polymerase. Adenovirus can use, right? Or it can use the viral, or virus itself can bear their own DNA dependent RNA polymerase. And then splicing, the splicing thing will be of course from the host, and 3' poly A tail processing, all these things, it is a host thing, right? And then it will make the viral mRNA. And for the from RNA to RNA, how it will work, let's say from negative sense RNA template. From negative sense RNA template, it has to make some positive sense RNA template, right? So the in our cell we don't have the RNA dependent RNA polymerase, but the virus has to carry the RNA dependent RNA polymerase, right? And yeah, so you can see here the virus is making some kind of cap structure as well. In case of influenza, they use host capping machinery, and also it snatches the cap from the host mRNA. That's why it's called cap snatching. But in other cases, virus also can carry their own capping machinery as well, right? So here is some example. Let's say if you have some negative sense RNA virus, that's RSV. First example, pneumoviridae family. First, if the virus want to make the protein, the virus first should make the mRNA, right? mRNA, and from the positive sense mRNA, it will be like it can make some protein through translation, and the translation will happen where? In the cytosol of host, using host cellular machinery, right? And let's say the virus now they have to replicate. How it can replicate? So it can make the complementary strand of this, and later again the virus, let's say this is the negative sense, and then it can make some positive sense, and later on it can use the positive sense as a template. Using the positive sense as a template, it can make a lot of negative sense RNA, right? That's its own genomic material, right? The same thing is shown here. If one virus has negative sense RNA, first it need to be positive sense mRNA, and following with this, all the translation, transcript, everything will happen. Yeah, this is the same thing that I explained, that if you have one positive sense RNA virus, first how it can replicate. If you have some positive sense RNA virus, some RNA dependent DNA polymerase, it will make some negative sense DNA of it will negative sense DNA, right? And using this negative sense DNA as a DNA dependent DNA polymerase, it can make some dsDNA intermediate, and from this dsDNA intermediate again you will get some positive sense mRNA. So if it's negative sense mRNA, what it will do? It will make some complementary. But if it's positive sense, because let's say it is positive sense, right? Positive sense RNA, it can make some protein. But for its replication, it needs some genetic material as well, right? So from there, first it will do negative senses single stranded DNA. From there, it will make double stranded DNA. And from double stranded DNA, it will make again plus senses RNA. That's the basic things the virus will do. And from here again the virus can make some protein, right? Protein. Okay, that's all about the virus replication and transcription that you need to remember in terms of GAT-B exam. Now I have some question for you that you can pause and answer. So RNA virus are entirely dependent on host cell. RNA virus are entirely dependent on the host cell for which of the following process? It's saying replication machinery, transcription machinery, RNA capping machinery, protein translation machinery. Pause, think. Okay, RNA virus are entirely dependent. It's replication machinery. Replication machinery, no, the virus can carry RDRP, RNA dependent RNA polymerase. Transcriptional machinery, the virus carry it, right? Let's say if it is a negative sense RNA, from there it will make some positive or intermediate, and after this intermediate again RNA dependent RNA polymerase, it can make negative sense. Capping machinery, maximum of the virus carry this except the influenza, but the protein translation virus can't carry actually, virus don't have. So for protein translation they must be dependent on the host cell. I am telling you multiple times, for protein translation the virus must be dependent on the host cell, right? Host cell ribosome, elongation factor, initiation factor, and termination factor. All these things they need. Next, RNA virus generally replicate in cytoplasm, nucleus, mitochondria, and late endosome. RNA virus generally replicate in where? That is the first slide. Think. So the virus is coming inside the cell, right? Now it's making some, let's say it's making some protein, the basic protein that's required initial, and then they are replicating in the cytosol. Only some viruses, as an example of influenza, they go inside the nucleus because of the cap snatching and the 8 segmented genome. The correct answer will be the cytoplasm. Right? Next, the following statement is not true for the receptor binding site of the virus. That means if it's the enveloped virus or something, it has some glycoprotein, surface glycoprotein, right? So the surface glycoprotein in one domain is called receptor binding domain or receptor binding site. So it's saying which one is not true. First, bind specifically to a cellular receptor. Of course it's true. This is true because you remember HIV has CD4, right? And then Epstein-Barr virus has some receptor, influenza has sialic acid, so they will, of course they have some specific receptor. Cannot bind to neutralizing antibody. What's called neutralizing antibody? Some antibodies produced inside the host when the virus infect the human or something. Those neutralizing antibody, the main neutralizing term came because they neutralize the viral action. They bind to the receptor binding site. That's why the virus can't actually bind to the cell. So this is not true. The neutralizing antibody will bind with the receptor binding site. Interaction of cellular receptor leading to activation of endocytosis. Of course it's true. It's composed of unique structural domain. It's also true. So the correct option will be cannot bind to the neutralizing antibody. It can bind to the neutralizing antibody. Got it? Next, some easy question. Which of the following virus shows a segmented genome and a cap snatching mechanism? Which of the following virus shows a segmented genome and a cap snatching mechanism? Pause and try. Right? Of course it's influenza. Segmented genome, 8 segmented genome, and also cap snatching mechanism. Next, which virus must carry its own RNA dependent RNA polymerase in virion? Which virus must carry RNA dependent RNA polymerase RDRP? Think. Let's say for here you need to think first thing, which of them is RNA virus? Poliovirus, is this RNA virus? Hepatitis C virus, is this RNA virus? Retroviral, which kind of virus is this? Right? It has some RNA from RNA to DNA. That means influenza. Of course, if RDRP is not there, what it will go inside the nucleus? Right? So influenza must carry the RNA dependent RNA polymerase. Viral host specificity is primarily determined by genome size, capsid symmetry, envelope glycoprotein, and viral polymerase. Virus host specificity is determined by which one? The thing is that where virus will bind, right? This virus has one surface glycoprotein and host has some receptor. Host virus. Of course, the surface glycoprotein. Through this, the bind host specificity will determine. Right? Correct option is C. Then, which step is unique to retroviral replication? This is very simple. Which step is unique to retroviral replication? RNA synthesis, integration into host genome, capsid assembly and budding. Of course, it's the integration into the host genome. From RNA, it makes some DNA, and then this DNA, this dsDNA and this dsDNA integrate with the host genome, right? So that's all. Okay, thanks, and today we will continue with some of the microbiology chapter again. Okay.